Okay, so in mitosis, the number of cells are diploid, and again in meiosis also they become diploid eventually. This is the last stage that we will discuss later. Okay, how do they become diploid, that we will discuss later. Now I hope you completely understand now what is diploid, haploid, and now see, we call them, the haploid cells are also called as daughter cells. Why? Because these are the resultant cells of the of meiosis. So they are in pairs. When they are in pairs, we call them homologous. Which cells are in pairs? Diploid cells, which we have seen in the diagram, that the diploid cells are present in pairs, and they are known as homologous pairs and homologous chromosomes also. While haploid cells, the cells are with the half number of chromosomes, and they are not in the form of pairs. Understand? So the word meiosis comes from the Greek word which is meion, means making smaller. Why? Because the diploid gets um, to take the form of haploid. It results in the reduction of chromosome number, but in the end, eventually, the number of chromosomes is uniform, it is maintained. How, that we'll discuss later. It is not reduced, but at some point it is uniform, and it is again the same. Okay, the same as mitosis. Again, they become diploid. How, that we'll discuss later. Okay, now the phases of meiosis is that it was discovered by German biologist Oscar Hertwig, and the year was 1876. The preparatory steps are identical to the interphase of the mitosis is identical to the preparatory steps of the meiosis. Okay, phases are G1 S phase, G2. After that, what happens? Meiosis 1 and then meiosis 2. Interphase is the last, interphase is the start, and then what happens? Meiosis and mitosis. Meiosis 1 and meiosis 2. See, these are very easy. Just you have to make your concepts clear. Read it again and again. Ask me if there is any issue. You will understand it completely. Okay. In meiosis 1, homologous chromosomes in a diploid separate in a diploid cell separate, and two haploid daughter cells are produced. In the step in meiosis, it generates genetic variations. How how do how does it generate genetic variations? How do we look different? How do we look different from each other? See, all your siblings, your brothers and your sisters, they are not the same photocopy of your parents. They are different from you, and they are different also from their parents. Why? The parents are the same, the daughter cells should be the same, so they should look the same. Because there is genetic variation, so that is why meiosis is significant from mitosis. Why? Because its significance is genetic variation. Because the genes are changed a little bit, the nature is different, the eye color, the hair color, the appearance, the height, everything is different. Okay, so phases of meiosis. Prophase is the first phase in the meiosis 1. Two main steps are there: karyokinesis and cytokinesis. You already know what is that. The karyokinesis is divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. This is the karyokinesis. So prophase 1 is the longest phase in meiosis. During this stage, the chromatin condenses into the chromosomes. The homologous chromosomes line up with each other and form pairs, which is known as see. This step was not present in mitosis. This step is known as synapsis. It is the crossing over. This is a very important point in prophase 1, and it is very important in terms of your examination also. Um, that this process is known as synapsis. The homologous chromosomes become bivalent. Again, see, bivalent. What is bivalent? Actually, I'll make a simplified diagram of this. So see, in this case, See, this is what happens in case of synapses. These are homologous chromosomes, means they are present in pairs. This is one homologous pair and this is one homologous pair. They join together in case of they are they what happens is that they change the genetic material. Okay, see here they line up with each other and they form different pairs. See, firstly they were present in this in pairs, like, which is shown by unicolor red, and here it is shown by blue. Now a different pair is formed and now they become bivalent. Again, two pairs are pairing with each other. What happens is that the genetic material is changed, which you can see here. The red is um, the red is transferred to the blue one and the blue to the red one. It is shown with the help of colors so that you can spot it out easily. What happens is that the genetic material inside the um, inside the nucleus, inside the division of meiosis, in the steps of meiosis, this takes place. Understand? This is not related to the fertilization. This is happening before the fertilization. See, I told you that the resultant is haploid. Haploid means single. Even they are not even single here. They are not even single here. But the process is not completed. This is the first step, and the synapses happens in the first step. They pair with each other. They swipe the genetic material, and now they are recombinant. Understand now what is recombinant? Because the combination has been done again. So re means again, recombinant. Now they are recombinant. Now they are recombinant chromatids. See again the same process, but here see, these are the homologous chromosomes. They become bivalent, two pairs. Then what happens is that the genetic material is swiped up, exchanged. Now they are known as recombinant chromatids. And actually, see, we call them one more thing, very important, this and this, because they are attached to the same centromere. These are sister chromatids. Understand? But the other chromatid of the other DNA, other pair, and the chromatid of one another pair, they are non-sister chromatids. Understand? So now you understand what are sister chromatids and what are non-sister chromatids. So see, these are these two are non-sister chromatids. Understand? But these two are sister chromatids because they are attached to the same centromere and they are a part of the same chromosomes. Homologous chromosomes. Now again, see, please don't get confused in the terminology. This is a confusing terminology but very easy to understand. Homologous chromosome means this one and this all this is a homologous chromosome. How? See, this is a homologous chromosome, this is a homologous chromosome. Why? Because they are paired up and they are homo means same. They are attached to the same centromere. They are similar to each other, so they are homologous. Now sister chromatids are present in a homologous chromosome. Do you understand this? See, here the sister chromatids, this is a sister chromatid and this is a sister chromatid. They are present in a homologous chromosome pair. And what are non-sister chromatids? Non these two are non-sister chromatids and they are present in non-homologous pairs, which means they are not similar to each other, they are not related to each other. Understand? Now, see, we call them as chiasma. Chiasma also. This changing of genetic material is known as chiasma. This is known as bivalent and it is known as tetrad also. Tetrad. See, this is known as tetrad also. Tetra means four. So it is also known as tetrad. It is also known as tetrad. Okay. After the formation of bivalent chromosomes and tetrad, same thing. What happens now? They are known as recombinant chromatids. Why not we call them as recombinant chromosomes? No. Why? because here only one only one chromatid is recombinant, and here again only one chromatid is recombinant. See, this and this, only this. Here this one is not recombinant and this one is also not recombinant, so we call them as recombinant chromatid. Now again, what is chromatid? We already know this is known as a chromatid. This single is known as a chromatid, which is not in the form of pair, and this is known as a chromosome. Understand? Which are present in the form of pair. Again, key concept: synapses is the connecting of homologous chromosomes. When the homologous chromosomes are connected, it forms what? We call them synapse. We call it synapses. They form what? Tetrad. Again, I told you about the tetrad. This process is the tetrad formation. What is crossing over? See, they can ask you about synapses, they can ask you about tetrad, they can ask you about chiasma, they can ask you about recombinant chromatids. They can twist and turn the question, but the answer is the same. These are different terminologies of a same process. The phenomenon is the same, but just the names and the terminologies are a little different from each other. So crossing over is the sharing of genetic material between two non-sister chromatids in a homologous pair. Now you get the definition of crossing over. It is the sharing of genetic material between two non-sister chromatids in a homologous pairs. Now key terms are tetrad and recombinant chromosomes. Now coming back to the uh, points which are mentioned in the book. This is your textbook. Now prophase. Now when does synapses happen? During prophase 1. Understand? The homologous chromosomes line up with each other. You know what is homologous. They form what? Pairs. You already know. They do what? They do for which process? Synapses. Now it is known as bivalent, and bivalent has 4 chromatids, so we call it tetrad. Again, what do we mean by 4 chromatids? Chromatid, again and again I am teaching you the same thing so that you don't get confused. What is a chromatid? This singular is a chromatid. Understand? This is a sister chromatid when it has a different sister chromatid also. This is a chromosome. This is sister chromatid, and this is also a sister chromatid. This all is a sister chromatid, and again this is also a sister chromatid. Understand? So why? Because they have 4 chromatids, so tetrad. The two non-sister chromatids of homologous chromosome, they join each other at certain points, any point, certain point, any certain point. They can join at any certain point. Where is the synapses taking place? In this picture, the synapses is taking place here. See, the genetic material is changed here, and now again recombinant chromatids can be seen. The points of attachment is now chiasmata is the point of attachment where they attach to each other is known as chiasmata. Where is chiasmata? It is here? No. No. Is it here? Where is the chiasmata? In our case, is it here? No. Is it here? No. This is the point of chiasmata. In the next stage, the non-sister chromatids exchange their segments and the phenomenon is known as crossing over. Chiasmata is the point of attachment. This is the point of attachment. Understand? Here, this is the point of attachment. This is chiasmata, and after the changing of the genetic material, we call them um, recombinant chromatids, and the process is known as crossing over. Now the exchange of segments results in the recombination of genetic information we call homologous chromosomes. Now they remain bivalent. Understand? Bivalent means they are not tetrad and they are not in the form of 4 chromatids. Again they become bivalent. Chromosomes condense further. What do you mean by bivalent? This is bivalent. This is bivalent. Understand? Please note it down. This is bivalent. This this is bivalent, understand? And what is a chromatid? This is a chromatid. This singular is known as a chromatid, and this is known as bivalent chromosome. Why? Because they are present in pairs. Chromosomes condense further, the nucleolide disappears. We already know the nuclear disappears, the nuclear envelope disintegrates, the centrioles which were present, they do what? Now they get duplicated, they migrate towards the two. Now further steps are the same, but the steps in the start, which is the synapses and the crossing over, this is different. Now it is the same process. Non-kinetochore spindle fibers, they interact with each other. The kinetochore spindle fibers will attach the centromeres, and in mitosis we have seen two kinetochore spindle fibers attached with one chromosome. Same thing happens here. It attaches with the help of a pair, which we can show in the diagram. If you see the diagram, you will see that the pair is attached. Understand? Metaphase 1, the pairs of homologous chromosomes align along the equatorial plate forming a metaphase plate, which we already know. Anaphase 1, kinetochore spindle fibers shorten. Pulling apart of chromosome is seen. One chromosome is pulled towards the other, haploid sets are formed. Same thing. Telophase 1, what happens during the telophase 1? Now this one, the American geneticist Thomas Hunt observed the phenomenon of crossing over in fruit fly Drosophila melanogaster. They can ask you the scientific name of housefly, like the fruit fly, which is Drosophila melanogaster. Okay, it is present. It is done. It is there in your book, so they can ask you anytime. Telophase, chromosomes arrive at poles. Each pole has half number of chromosomes, but each chromosome is still consists of two chromatids. The number of chromosomes is half, but again two chromatids are there. Spindle network disappears, the nuclear envelope is formed, and they uncoil back to the chromatin. Cytokinesis, you know already, is the pinching of the membrane and the formation of cell wall in plant cells. It forms the cytokinesis also forms the daughter cells, the cell wall in plant cells. Okay, during the crossing over, the genetic material is exchanged between sister and non-sister chromatids of homologous and non-homologous chromosomes. Do they interchange with sister chromatids? No, they always interchange their genetic material with the non-sister chromatids. After meiosis, both the haploid daughter cells enter a period of rest, which is known as interkinesis and interphase 2. The interphase 2 is different from the interphase of mitosis and meiosis. There is no S phase, there is no duplication of chromosome during this stage. Okay, after meiosis, the haploid daughter cells enter a period of rest, which is known as interkinesis or interphase 2. The interphase 2 is different from interphase of meiosis and mitosis. There is no S phase and there is no duplication of chromosomes. Understand? So after telophase, there comes the interphase 2. So what happens during the interphase is that the haploid cells are now produced and now they are resting, and there is no S phase, so there is no duplication of the chromosomes. Now no duplication will take place. Okay, now we can see the differences between the differences between the mitosis and meiosis. Okay, uh, no, that will be done later. These are the stages of meiosis. So the first stage is the interphase. Now after the interphase, the second stage is the prophase. What happens during the interphase? Chromatin, you can only see the chromatin and the centrosome. The chromosomes are not visible. In the prophase, the chromatin gets condensed. See, this is the chromatin, it gets condensed, and no, here this will be clear for you. This is the chromatin and this is the spindle formation. Now chiasmata formation, as we have already studied in prophase 1, the chiasmata formation is seen. Then metaphase plate is there in the metaphase 1. Why do we keep calling 1 1 1? Because prophase 1, metaphase 1, because these are all the steps of meiosis 1. Now anaphase and telophase. These are the steps of meiosis. One, please keep in your mind it is similar to that of mitosis, other than chiasmata formation and the crossing over. Other than the crossing over, all the steps are same. Anaphase, in anaphase, chiasmata gets broken, and now you can see you have to, whenever you are making the cycle of meiosis, you have to denote this with the help of your pencil. You can shade one area and you can leave the other area unshaded. You cannot use colors, because you can make it with the help of a like black pencil. Why? Because you have to show the process of crossing over. If you are not showing that, this can reduce your marks, because in meiosis the process of crossing over does happens, and this phenomena has to be shown in the diagram. Then comes the telophase. Each daughter cell has haploid number of chromosomes. See, where is the haploid number? You see, what happens is that this will get separated from each other in meiosis 2. Now you will study the other steps in meiosis 2. After telophase, what happens is that it goes into a resting phase, which is known as interphase 2. See, why interphase 2? Because this is interphase, first interphase. Okay, then comes prophase 1, then metaphase, which we have already studied. After metaphase, anaphase, telophase, again interphase 2. Why 2? Because now they are again going into the interphase, which is the resting stage, and after the it goes into the resting stage after some time, then meiosis 2 takes place. Okay, stages of meiosis 2. Again, in case of meiosis 2, you can see another prophase, which is prophase 2, then metaphase 2. This is the prophase 2, this is the metaphase 2, this is anaphase 2. Why now? Why 2? Because we have two daughter cells, and the division happens in case of anaphase and then telophase. Now you can see haploid daughter cells. Each daughter cell has haploid number of chromosomes, and again they are showing the crossing over with the help of differentiation in the colors. What is meiosis 2? It is the second part of meiosis, and it is similar to mitosis. So the first part is different. Why? Because of the crossing over. The second is similar, which is meiosis 2 is similar to mitosis. Meiosis 2 has the same steps which are similar to mitosis. Why? Because there is no crossing over, no recombinant chromatids, no synapses. Okay, it is divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 2 takes much less time as compared to prophase 1. In this prophase, nucleoli and nuclear envelope disappear and the chromatin condenses. In case of prophase 1, see here, the prophase 1, see the nuclear material, this, where is the nuclear envelope? You must know that this is the nuclear envelope which disappears and the chromatin condenses. See here, chromatin again, it condenses and the nuclear envelope disappears and it spreads inside the whole cell. The chromosomes, then they form a metaphase plate, and then they spread. Okay, centrioles move to the polar regions and make spindle fibers. In metaphase 2, the chromosomes attached with kinetochore spindle fibers and align at the equator of the cell. Then anaphase can be seen, anaphase 2, where centromeres, then they same, it is it is the same as mitosis. The centromeres get cleaved and then they are pulled apart, the chromatids are pulled apart, the sister chromatids are known as the sister chromatids are now called sister chromosomes, and they are pulled towards opposing poles. And telophase 2 is marked, it means telophase 2 is related to the uncoiling of chromosomes in chromatin. Nuclear envelopes form again. See, I keep on giving you substitutes of different words. If you forget reform, you can say nuclear envelopes form again. Cleavage or cell wall information eventually produces how much daughter cells? 4. With a haploid set of in one cycle of meiosis, how many chromosomes, how many daughter cells are produced? 4, which with haploid set of chromosomes. Okay, Now comes the significance of meiosis has been omitted, but you must know how does it maintains the number of chromosomes in the next generation. See, because meiosis, this is omitted, but uh, I'm going to I'm going to teach you this for important information, okay? Because this information is important, we must go through this. Why? Because see, the male gametes again haploid, the female gametes again haploid, the sperms and the ova. When they join together, they form what? Zygote, which is known as actually the fertilization. And in case of zygote, repeated mitosis increases the number of cells, and the growth of zygote is also seen in the form of embryo. Okay, now see, the most important thing which I wanted to let you know is that the chromosome number is maintained for the next generation. How? See, I'll tell you. This is one chromosome, one chromatid haploid. This is male, and this is one chromatid diploid, which is female. This is from female, this is from the egg, and this is from the sperm. When they are going to join with each other, what happens? A zygote is formed, and then again this zygote has double number of this has a pair of chromosomes, and again same number. See here, as we all know that they have how many sets of chromosomes? 23. Again, when they join together in the form of embryo, they again have how many? 46. So Allah Ta'ala has kept a process for the maintenance of chromosome number. It has to be maintained in the it has to be maintained in 46. It has to be maintained in the number 46. How? After fertilization, again 46. In germ cells, in gametes, 23, but after fertilization, 46. What happens if it was 46 already? What will happen? Okay, so see, in case, okay, now if the chromosome is, if they are already 46, understand, Allah Ta'ala has made a process which is meiosis. Why? Because we want to convert them into haploid. If they are not one, if they do not become n, see, the proper process is that they are present in the form of n. See, one is from the female, it is n. Again from the male, again it is n. And then when they join together, what happens is they become 2n. We need to maintain this nuclear material, which is 2n, which is diploid. Understand? If it is already 2n, see, if the number of chromosome here is 46, if it is already 2n, see this is if I'm talking about if it is already 2n, 2n, and this one again, this is 2n, what happens in the as a result? They will become 4n. Are human beings 4n? No, they are diploid. We don't want them to become 4n. That is why Allah Ta'ala has designed a process, and this process does what? This process makes haploid gametes. Haploid gametes, haploid gametes. So when they join together, they form diploid. Again, what is diploid? They become in this they become in this form. Understand? Homologous chromosomes form. This is chromatids, this is chromatid, and these are chromosomes. Understand? So if they are not half, understand, if they are here, they are n, they are here n. After, in the end, this is n, and this is also n. In the end it becomes 2n. Understand? So we have to maintain this diploid, we have to maintain this double nuclear material, we have to make it diploid. Why? Because all the cells of the newly formed embryo, because see, any baby when it is getting when after the fertilization, when it is growing, when the cells are growing in number, if they are haploid, what will happen? The human or baby of any animal will not be normal because the chromosome number is changed. We don't want the chromosome number to be changed. So Allah Ta'ala first makes it half n and then makes it 2n. Understand? When 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 the gametes fuse. When do they fuse? When the ova and the sperm fuse with each other, and this is the process of fertilization. Understand? These are the errors in meiosis. They're not a part of your syllabus. So the comparison between the meiosis and the mitosis. Meiosis 2 is similar to mitosis, while meiosis 1 is actual difference between mitosis and meiosis. Meiosis 2 is similar to mitosis, but meiosis 1 and mitosis are different. Why? I already told you, I have already mentioned, because of crossing over. The following chart describes the main differences, so we have to go through this chart. See the here, first of all, the interphase similar. The interphase in the mitosis and meiosis is similar. Now the prophase is different. Why? Because the homologous chromosomes do not form pairs. There is no crossing over in mitosis. Here they form homologous. Here they form pairs. And why pairs are formed? For homologous, between the homologous chromosomes, because the crossing over is there in the homologous chromosomes. That is why the prophase 1 is different, and that is in case of meiosis, not mitosis. Metaphase, single chromosomes align to form metaphase plate, and here homologous, see, homologous chromosome, see the tetrad, remember the tetrad, here they are 4 and here they are single. Understand? Now singular chromosomes are there to form a metaphase plate in mitosis, but in case of meiosis, the metaphase plate is formed by homologous pairs, like actually tetrad 4. Remember, bivalent. Bivalent means two pair of chromosomes, actually, which becomes 4 chromatids. Understand? Now, in case of anaphase, the chromosomes break and individual chromatids are pulled towards the poles, but here the individual, here see, here they are talking about chromatids. See, here the chromatids are going to get pulled, here the chromosomes are going to get pulled. You can see here the difference. This is like 4 and 2. See, these are the chromosome. This is one chromosome, this is one chromosome, this is one chromosome, and here singular chromatid is present, singular chromatid. Understand? Daughter nuclei contain haploid number of chromosomes. Each chromosome has 2 chromatids. Again 2 chromatids, but here daughter nuclei contain diploid number. See, here they are diploid and they have single chromatid. Understand? We are talking about mitosis and meiosis. 2 chromatids are present here. Here we have 2 chromatids and here we have 1 chromatid. Sorry, here we have 2 chromatids, 1 chromosome, homologous chromosome, and but in case of mitosis they are present as single chromatids. So please don't get confused. Here they are double and here they are single. Don't think that meiosis has single, so you make single here. No. In case of meiosis 1, we are talking about meiosis 1, but the meiosis 2 is same as that of mitosis. Understand? So that is why they have not discussed meiosis 2 here. They are only discussing the differences between mitosis and meiosis, which means mitosis versus meiosis.